Crystallization of pharmaceutical active ingredients, particularly those that possess multiple polymorphic forms, are among the most critical and least understood pharmaceutical manufacturing processes. Many process and product failures can be traced to a poor understanding and control of crystallization processes. This can have a significant impact both on product quality and downstream process units operation, including filtration, drying, milling, and product formulation. This presentation reviews typical problems encountered in production crystallization with case studies, advice, and strategy to understand and avoid those problems through the use of in situ crystallization characterization tools. Before to present some example, I would like to spend a few words about Redox. Redox is an CRO labs where are working more than 28 persons, most of them graduated. Redox is certified by several authorities for GMP, GLP, Food and Drug Inspected, narcotic analysis, REACH, and many other aspects. We Redox give a lot of support for many pharmaceutical companies in Italy and worldwide. Redox is organized in several departments. One department with the chemical physical analysis, the second department is solid state studies of API and development processes. There is also a very strong department where are carried out safety evaluation for the chemical process in support to the development scale. The last department, there is also impurities, isolation, and chemical processes development. Concerning this department, all the people are involved in a specific activities. Between all the activities available in Redox labs, I want now to present some examples carried out in the Crystal Department group in support to the crystallization processes quality requested from our customers. In this slide, I summarize all the subjects I want to present during my speech. I will talk about the crystallization strategy for quality, talking about the critical quality attribute of the product, the critical process parameters of the plant, the analytical control procedure, mainly online, how to determine the metastable zone width, and following there is some example on research scale and on the scale-up strategies. Before to start the crystallization development strategies, it's important to take account that two main points should be evaluated. First of all, explorations, and the second point, exploitations. Exploration means based on the existing knowledges are not sufficient to solve the problem identification, new technology are needed to create and acquire to contribute to existing body of knowledge. At the same time, a continuous contact with exploitations means utilization of existing knowledges for innovative problem solving. These two key points must be kept in mind to develop new strategies. Anyway, before to start any crystallization strategy for the quality, must be keep in mind the two main points. One is the critical quality attribute of our products, and the other the critical process parameters. Critical quality attributes are considering the particle size, the polymorphism, the solution stability, while the critical process parameters are considering all the parameters involved in crystallization. For instance, temperature, time, stirrer speed, variables, loading reagents. Certainly, no critical process parameter must be taking count if there are no probability to have a failure during the crystallization processes. In the pharmaceutical context, in order to get a very good stable compound under specification GMP, good manufacturing practices, it's important to keep in mind several points. First of all, to understand the thermodynamic properties like polymorphism, suspension, crystal surface and water energy and so on. Other points are the kinetics related to nucleation rates, crystal growth, or solvent properties on the physical chemical systems. In parallel, to get the good crystals, it's important to know the operation condition. In other words, if we look for the very stable polymorphic form, crystal shape, purity, all these parameters must be considered. Before to present some examples, it's important to keep in mind what does mean crystal properties. Crystal properties can be looked at by internal properties, polymorphism, dissolution, and whatever, or by external properties, morphology, particle size, aggregation, and so on. Certainly, it is needed to apply analytical control methods to evaluate these two different approaches. One is online methods, by which is possible by particular equipment to define the particle size like focus beam reflectance measurement, or the crystallization procedure by calorimetry, or by spectroscopy, FTIR or Raman, or by 2D imagery. Other analytical methods should be carried out by offline methods like particle size diffraction methods, X-ray, scanning electron microscope, optical microscope, and so on. The online give in a real time the crystal properties during the crystallization. The offline are more precise methods, but they have some limits because they need long acquisition time. Sometimes the sampling are creating process interaction during the sampling, and these are crucial for evaluation. The first example now I am going to present is related to the application of reaction calorimetry, focus beam reflectance measurement, and FTIR in support to the crystallization process in which a product can give a different polymorphic form, different morphology, and also particle size, different distribution particle size. This active ingredient can crystallize in two different polymorphic forms. As shown by DSC heating curves, it presents a form 2 melting at about 110 degrees, followed by the polymorphic form melting point at 140 degrees related to the form polymorphic form 1. In relation to the crystallization processes, it's possible to get a mixture of one of these two pure polymorphic forms. In relation to the process conditions, it's possible to get a different polymorphic form with different morphology. Form 1, as shown by photos, is characterized very well-shaped particles with very well-defined morphology, while Form 2 presented an aggregate. An additional problem was also related to the different flowability of these two parameters in relation, of course, to manufacturing processes. The first step of investigation was to carry out in the lab scale these crystallization processes in a reaction calorimeters equipped with the focus beam reflectance measurement probe with FTIR ATR, and then collect the samples following the customer procedure. At the end of the test, carried out with not control process, some analysis has been carried out, DSC, particle size, and scanning electron microscope. By DSC, it was possible to see that the product is practically a mixture of the two polymorphic forms. By scanning electron microscope, it was easily to see that the photos show aggregate, not very stable and homogeneous particles. At the end, the particle size distribution presented distribution curves very broad in very large interval of particle size. By calorimetric profile obtained by RS1, the only D process, it was possible to point out the crystallization profiles. That it's a very crucial point to show how this crystallization can occur. In fact, if you look at these plots in which are reported the temperature recorded starting from 60 degrees until 40 degrees, and then after the plateau to reach the 0 degree, about 0 degree temperature, it was possible to record two different peaks, exothermic peaks. The first one with a sharp peak at about around 40 degrees, and later a second exotherms along the cooling rate from 40 to 0 degree, in which other crystals have been obtained. Looking through the heat curves, it was possible to obtain the conversion in terms of calorimetric conversion that figure out how much the heat percentage in relation to the temperature was obtained. Two different plateaus. The first one from the 40 degree up to the first point, and then the second one in the percentage at about 20%, 30% from this during the second steps. Parallel, of course, was possible by FTIR ATR probe recorded the concentration profile versus the calorimetric conversion profile, and also in relation to the percentage of crystallization products. In fact, in relation to the temperature line, starting from 60 degree up to 40 and then to 0 degree, there is a decreasing of the concentrations recorded by FTIR, ATR, and practically by conversion profile, there was a very clear percentage correlation between the heat recorded by conversion calorimetry. At the same time, with the same experiment, not only crystallization process by calorimetry and concentration variation was recorded by FTIR spectroscopy, but even by focus beam reflectance measurement probe was possible to obtain plot related to the number of particles of crystal formation during the calorimetric conversion, during the temperature ramp. And practically, a good correlation was observed by calorimetry and the calorimetric profile and the number of particles in terms of number of crystallization compounds. What was decided to do? After the first experiment, a design experiment in terms of crystallization processes were put in place. Several trials were carried out. First of all, were defined the metastable zone, and then were changed the temperature profiles and the induction time for the crystallizations of the polymorphic forms. In this plot, we report an example of different approach by which was changed the parameters. And practically, this plot presents the calorimetric profiles in terms of heat flow during the crystallization, by which changes the temperature from 63 degrees up to 42 degrees prolonged the time of nucleation, and practically was possible to get a large peak of exothermic related to crystallization of foam 1, and after the plateau at this temperature, 45 degrees, reduce slowly the temperature up to 10 degrees and obtain a very short, a small amount of another exothermic. This profile, heat profile, was converted in conversion heat flow and give us a lot of information how to change the crystallization processes. As shown before in the first experiment carried out with a normal process, in this case was also recorded the concentration by FIR spectroscopy versus calorimetry, and the profile of FIR give a different behavior and a lot of information how to better perform the crystallization process in order to get the better forms, better crystals, better polymorphic forms. Then the analytical data recorded on the solid isolated with the new methods obtained after design experiment trials give this data. The SE show that the product is practically complete crystallized in form 1 with a very low amount of form 2, the blue figures. The particle size was reduced in number of distributions with the maximum percentage below 170 microns. And first of all, the shape of the particles are completely different than aggregates. The crystals are more homogeneous and better to manipulate, to manage in formulation also, and in relation to the flowability processes. The second example I am going to present is concerning the study we have carried out to define the perfect nucleation process conditions in relation to the critical process parameters during the crystallization of an NAPI. And that was especially focused in defining the development stage in support to the quality specification required from the final product. Final specification concerning the define a pure polymorphic form, define specification with a particle size distribution specific requirement, and the last but not the least, the particle morphology that has a big impact in flowability of the final product during the formulation, during the transfer for tablet formations. Also in this case, to evaluate the process, we use the reaction calorimeter equipped with the focus beam reflectance measurement probe. In this first slide is reported what we have recorded during the first trials following the normal process conditions. Red line means the temperature of reactors, blue line the heat flow. After the endothermic solubilizations, during the cooling step from 80 degrees up to 42 degrees, an exothermic crystallization peaks with the 2 peaks were recorded. This is practically the process that is present in the production scale. Several trials were performed, and like a design experiment, were obtained different behavior, crystallization behavior. I show you in this slide one of these trials, the third trials, in which after the solubilization of the curves recorded by reaction calorimeters during the cooling profile in which the temperature was changed at about 46 degrees, the exothermic crystallization presents only one peak, large peak, with a different behavior, different profile as we have seen in the first experiment. Of course, these exothermic give us a lot of information about the induction time for nucleation and give us also the information how to manage the process, how the critical parameters of the process can influence the crystallizations. Of course, after this experiment, other data were obtained by focus beam refractor measurement and also by collecting the powder after crystallization with the offline analytical method like X-ray, focus beam refractor measurement, and so on. During the first trial, by a focus beam refractor measurement, it was possible to control the particle size distribution when the exothermic event occurs. In fact, by the curves, blue curves, yellow curves, green curves, it's possible to see the different fraction of particles occurring during the crystallization that at the end give a particle size distribution characteristic of two different populations. While in the third trials, where exothermic event, homogeneous exothermic occurred, only one population of particles was obtained, and that was confirmed also by particle size analysis offline. Well, if you compare the distribution profile of the particles isolated from trial 1 and trial 3, we can see that there is a good correlation between the shapes of the calorimetric peaks and the particle size distribution. This is the good correlation that gives us a good idea how to manage the product, how to get a perfect crystallization processes in order to avoid any undesired process. Well, from the exothermic profile, where are two peaks? Are these two peaks crystallization process related to the different polymorphic form obtained in relation to the different temperature? Well, other investigations were carried out on this product, and from different studies, it was seen that this product presents polymorphic form, two polymorphic form, form 1 and form 2, as shown by X-ray powder diffractions. While looking from a mixture of the experimental 1, where I compare X-ray powder diffraction of this solid isolated from the experimental 1, it's possible to see that the two peaks related to exothermic event recorded are related to the mixture of the different crystallization forms, forms 1 and forms 2. In fact, from the black line, black peaks, the diffractograms are present peaks related to the form 1 and form 2. All these are really shown by the error of the peaks related to the other polymorphic forms. Well, in addition to the different polymorphic form, there were another question to answer. Why in one case there were good flowability in terms of transfer product in relation to the process? Well, concerning the crystal morphology obtaining by photoscanning electron microscope, form 1 present a standard form, present a plate, very homogeneous plate, while form 2, a pure form 2, is present at the solid state as a needles. Certainly, needles can create troubles during powder flowability, during the loading machinery, and so on. Well, if you compare the scanning electron microscope photographs collected from the experimental one, we can see there are not homogeneous products. And even in the second experiment, not described in detail before, are present of different polymorphic form. Practically, this different behavior is also related to the different polymorphic form. That was clearly shown by scanning electron microscope. Well, while in trial 3, where was obtained only form 1, the specific form, desired form, the shapes of particle size and the morphology are related to a good and expected specification for this product in relation, of course, to temperature, in relation to induction temperature and the crystallization processes. And practically, what we can see that by definition of the critical attributes, by using different techniques, online methods, for instance, RS1, calorimetry, focus beam reflectment, in combining with the other technique, offline technique, like polymorphic forms defined by X-ray powder diffractions, focus beam reflectance measurement, and other particle size probability test, it was possible to give an answer with a very limited number of experiments and give a good approach for scaling up the process. In conclusion, which kind of message I want to present with my examples? Concerning the crystallization in terms of good manufacturing practices and regulatory rules, the quality must be evaluated through the crystallizations, through the solid form screening selection, by in-process analytical methods, suitable methods, online methods if available. All these in relation, of course, to the formulation processes, in relation to the final dosage product. That is even requested by Food and Drug Administration regulatory, by national and international rules, and these should be followed before to start any crystallization of active ingredients. At the end of my presentation, I would like to thank the professionals that are working with me and carry out the experiments in the labs, controlling the reactions in the bioreactions, like Nicola and Davide, or the other person, professional that carry out the out-of-line analysis, like microscopy, like X-ray powder diffraction, particle size, Marco and the other Marco. Both are full-time engaged for the solid state in Redox. Thank you for your attention.